My name is Brian Morris. I'm Professor Emeritus in the School of Medical Sciences at the University of Sydney. I've been chosen for the Irvine Page Alva Bradley Lifetime Achievement Award at this year's meeting in San Francisco. I became interested in the area of health sciences generally as a teenager. I was quite a good student, and I often indulged myself by looking outside the curriculum. And I went to a talk by a chap who was spruiking his book. His name was Lee Lord Cordell, and the name of the book was Eat Right and Live Longer. This was held at the Adelaide Town Hall, where I grew up. Adelaide is the capital city of South Australia, one of the Australian states. Anyway, this really attracted my interest, the whole idea of diet and exercise. And so not surprisingly, I went on a track of not only applying these messages to my own life personally, but studying the medical sciences. I graduated from the University of Adelaide with honours, and during my final year, I did a project just by chance, starting in the summer vacation, in a lab that was doing renal physiology, where there was a discovery of an inactive form of renin. And there was a mystery. This was Eugenie Lumbers who discovered this. She had just finished her PhD and was about to embark on a postdoc a year later. So I spent a year working with her. And I came up with the idea that renin is like pepsin. It's part of the same family of proteases. And it's known that pepsin can auto-activate. That is, you can add pepsin to pepsinogen and it will activate. So I thought, well, let's try adding pepsin to renin and see what happens. Well, this inactive renin, really. And lo and behold, it activated. And quickly led to the name of inactive renin, as in prorenin, a precursor. So after my time in Adelaide, I was looking for a PhD, and Colin Johnson in Melbourne was interested in whether there was a renin-angiotensin system in the kidney. So I moved to Melbourne and embarked on this project, along with a number of other projects. And they've been trying to measure angiotensin in the kidney. But angiotensin is only a small peptide and rapidly broken down. So after about a year, I thought, well, why not see if we can measure angiotensinogen in the kidney? And that's exactly what I did and succeeded with, and presented Colin with the results. And he was quite happy. I also presented him with the manuscript at the same time, and we sent it off and it was published. And this really began the field of the intrarenal renin-angiotensin system, which now is known to be an important factor in hypertension. And many researchers over the years have confirmed this and expanded this concept, and it's now a well-accepted phenomenon. After I graduated from my PhD, I won a prestigious C.J. Martin Fellowship, of which there were only 2 in Australia that year, and they were for study in another country. And of course, the US attracted my interest because of the very strong research in the United States. And I went to the lab of Jim Davis in Columbia, Missouri, and did some physiology for the first time in my life, physiological research working on dogs, rabbits, rats, and I learned a lot. Hypertension, congestive heart failure. And after that, I did a second postdoc with Fran Ganong in San Francisco at UCSF, which by coincidence is, of course, where the meeting is being held this year. And that too was productive, and I had pretty much free rein to do what I liked. And so, as well as studying extrarenal renin-angiotensin systems, I did more work on prorenin and published more papers on that. After 3 years in the US, I was offered an academic appointment at the University of Sydney which I accepted, and set up my own lab. Difficult, of course, as everyone knows who sets up their own lab. Frustrating to get funding support and so on, but by various means and a bit of lateral thinking, I was able to raise the funds and get the lab going. And I quickly started to apply some of the other things that I'd learnt when I was in San Francisco. Because in the mid-70s, when I was in San Francisco, we saw the advent of recombinant DNA technology. And that really got my interest. And I became, in fact, friends with a lot of the people involved in that technology, people like John Shine, who was a fellow Australian, Joe Marcial from Belgium, Peter Seberg, Axel Ulrich, and John Baxter, who in fact in later years put quite a bit of money into my research in cloning the human renin gene. But before that, I actually elucidated the biosynthetic pathway of renin using some of the novel approaches at the time, and this confirmed for the first time the synthesis of pro-renin directly, and that was quite an interesting and important first. But the next one, of course, was cloning renin, firstly from the mouse, and then using it to probe the Maniatis library, which I collected from Genentech in one of my visits to San Francisco, taken it back to Australia. And this, in fact, was used by many people over the years in Australia to clone multiple genes, including my lab, where I cloned the first human kallikrein gene, and cardiac myosin genes and others as well. The work I did on cloning the human renin gene pivotally involved John Shine. He had a big recombinant DNA lab. Mine was more of a startup lab. It's the first in Sydney, but John's was the biggest in Australia. And of course, as I said, John Baxter funded the work, which is great. Sad to say, John passed away a year or so ago. So then I moved on, having cloned the human renin gene. I thought, where can I go with this? And I thought, wow, people have often wondered if renin is involved in hypertension, in the genetics of hypertension. So I carried out the first experiments to test for polymorphisms in the human renin gene for association with essential hypertension. It was negative, but it was a first. And that led me then into that field, and I performed lots of other association studies over the years, lots of other work, including genome-wide linkage study, which identified several loci, and then moved on to studying the expression of messenger RNAs across the genome, as well as microRNAs. Introduced quite a number of concepts over the years, which I'll talk about in my lecture. And what I found is that this brought me to a point where my whole life's work seemed to come together, because the intrarenal renin-angiotensin system that had been implicated in hypertension really didn't point to an exact mechanism involving renin. And what I found is that two of the microRNAs, well, there were two microRNAs that I pulled out of many microRNAs in the human kidneys in hypertension, that in fact were able to target the renin messenger RNA, specific target sequences in the 3' untranslated region, which is how microRNAs regulate the expression of genes at the post-transcriptional level. So we did some experiments in cells, cell cultures, transfecting them with constructs, and showed indeed that these microRNAs do have this effect on these particular sequences and destabilize renin messenger RNA. So nicely accounted for the higher expression of renin that my PhD student Francine Marquez from Brazil, brilliant student, showed. That was only a couple of years ago, published in Hypertension. And nicely accounted for why this downregulation of the microRNAs led to the upregulation of renin expression and therefore contributed in that way to hypertension. And the work is ongoing. It's now been confirmed with collaborators at the Baker Institute and with Francine, who's now at another university. Together, we've all worked together to find, in fact, and show that this same mechanism, in fact, one of these same microRNAs is present in the mouse and actually correlates with the blood pressure in these mice. So we see the same phenomenon of renin overexpression in the kidney as in this genetically hypertensive mouse strain, low microRNA expression and hypertension. And subsequent work is further confirming this in humans with other colleagues in the UK and elsewhere. And the mechanism is now becoming even clearer. It's a little bit different than involving the classical juxtaglomerular cells, but I won't say any more because all of that is still ongoing, but it's turning out to be fascinating. So, just to summarise, all I can say is thank you to the committee who selected me, thank you to Kurt Sigmund in particular for nominating me, and for those who I've heard have seconded my nomination. I'm absolutely thrilled, humbled, and overjoyed, in fact, to receive this award today. And I look forward to many more fruitful collaborations in the future. My current research has expanded enormously since then, and even though I continue to have hypertension as my top priority, I'm quite widespread in my interests, and in fact it covers paediatrics to gerontology and plenty of bases in between. And currently I'm collaborating, and I'm going to show 2 posters at the conference on Thursday of work that I've been doing with the Honolulu group during a sabbatical a year and a half ago, and with ongoing collaboration with them. And that's quite interesting. It's on the molecular genetics of extreme old age. So if anyone wants to see some of my other work, they're welcome to come to my poster on Thursday afternoon. Thank you very much.